Welcome back to this video blog on Alzheimer's disease. Now, you may have noticed a video blog with the same title on our website and our Facebook page. However, we were not happy with the quality of the video blog, so here's a enhanced, improved, and slightly revised version of the same talk, which we believe is important enough to give you a good quality product. So the title of this talk is Alzheimer's disease an infectious disease, which of course is a quite provocative title with fairly scary implications. We will break this talk down into several parts. So let's launch into part one, called amyloid. So as many of you may know, the amyloid protein is kind of the evil perpetrator in Alzheimer's disease, as it tends to clump together in neurons with adverse consequences, such as the later production of a protein called tau, which becomes hyperphosphorylated and causes the so-called neurofibrillary tangles, whereas amyloid causes the amyloid plugs. So amyloid seems to be the starting point of disaster in the aging brain. Let's dissect a little bit what amyloid actually does, where it comes from, and the other side of amyloid, which may have been overlooked thus far. So the first slide is amyloid disease and infectious illness, part one, amyloid. And the next slide launches us directly into the biochemistry and molecular biology of amyloid production. So as you can see here, amyloid is a huge protein inserted into the bilayer membrane of neurons and other tissues. And you can see here that amyloid can be cut and cleaved by a series of enzymes which release a so-called amyloid beta protein. Here, which by virtue of its biophysics tend to accumulate or aggregate into so-called oligomers. And it is thought that these oligomers are toxic to neurons and cause all the damage that is involved in amyloid deposition. Now the next slide gives you a more differentiated view of what's happening in the processing of amyloid. And you can see the complexity has increased immediately. You can see here on the top of the slide that there is a number of enzymes involved in the cleavage of the amyloid precursor and in the production of the amyloid beta proteins. At the bottom here are some factors that are extraneous or outside factors involved that are not directly linked to the molecular biology. And you can see here that aging, ischemia, diabetes, traumatic brain injury are all mentioned. And I've added here in a red box blood-brain barrier problems. And I will explain to you later as to why these might be very important. So just to recapitulate, the amyloid beta monomer is being produced here, and we find the aggregates, the oligomers, accumulating and then producing down the road, downstream, a insoluble tau protein, which is thought to be the trigger for neurodegeneration. Let's move on to the next slide. And here's another way of presenting this. So you have the normal protein, which can be misfolded in some way, lead to aggregation to oligomers. Oligomers may fall apart and seed other monomers, which again form oligomers, and this is then a process which can migrate through the brain. In a sense, it's been speculated that the amyloid protein may be a prion-like protein. Prions are infectious agents which do not have a genetic core, no RNA, no DNA. It took decades to convince the community that these were for real, but they now have been well established by Dr. Prusiner at UCSF, who a few years ago won the Nobel Prize for this work. So prions now seem to be a generally accepted concept. They are proteins which are infectious and by virtue of their biophysical qualities tend to interact with their brethren proteins in the infected organism and lead to a chain reaction of mass agglomeration of these proteins and damage to the brain. Some diseases involved here are scrapie and mad cow disease, of which there was an epidemic in Great Britain not too long ago, causing a great deal of concern. The next slide shows you that the propagation of the pathology does follow a certain path. Namely, it appears that, for example, tau tends to begin in the locus coeruleus, or very close at least to the entorhinal cortex. And we will get back to this structure in a minute when we talk about some other aspects of amyloid, and then has a progression into other brain regions. Recently, there has been a great deal of work detailing how these pathological protein aggregates may trigger aggregation of proteins in neighboring brain areas, and there may be such a thing as transsynaptic transport of the pathology of these protein aggregates. Next slide shows you that you can in fact produce an amyloid phenomenon in a brain of a mouse by injecting into the peritoneum, the abdominal cavity basically, beta amyloid peptides. These are transported in some way to the brain, may enter the brain, and cause a chain reaction very much in the prion concept and lead to amyloid deposition in the animal's brain. Next slide is now the consequence of amyloid deposition in the brain, namely the activation of microglia. You can see here that amyloid oligomers are interacting with receptors of microglia cells. These are so-called toll-like receptors. The microglia can be internalized. The AP molecule can be internalized into the microglia and then cause a chain reaction of cytokine release and an inflammatory response that is commonly seen surrounding amyloid deposits, which are thought to be one of the driving forces of neuronal pathology and neuronal death. The next slide shows you a cartoon which highlights this. So you have the microglia, which interacts with amyloid beta deposits. Microglia is now activated and will attack neurons and leads to damage neuronal cell death, as well as impaired synaptic remodeling, which microglia is usually involved with. The next slide shows you some pictures which prove this concept. So in panel A, you see microglia in blue directly adjacent to a beta amyloid deposit in the parietal cortex of a brain section of a patient with Alzheimer's disease. In panel B, you see activated microglia in green and an amyloid beta plaque site in the brain of a transgenic mouse which carries the APP, abnormal amyloid precursor protein gene derived from man. And in panel C, you see an astrocyte, one of the glia cells surrounding a amyloid beta deposition site. And finally, in panel D, you see that astrocytes are at a amyloid beta site in the brain section from another transgenic mouse. So evidence then from a variety of sources, both human brain, postmortem brain, as well as transgenic animals expressing or super expressing amyloid precursor protein and the amyloid beta protein, that microglia is intimately affected, and an inflammatory response universally results from the deposition of amyloid in the nervous tissues. The next slide brings us now to part 2 of this talk, and this is the big surprise: amyloid as an antibiotic. Now, it's been known from other species that small peptides can have antimicrobial effects. And some research became very curious about this, especially a group of in Harvard run by a famous Alzheimer's researcher, Dr. Tansey, who spent quite a bit of effort on this, and in the next slide have shown that brain homogenates of Alzheimer's disease patients, which by definition contain increased amounts of amyloid protein, are able to decrease the replication rate of Candida albicans in tissue culture. Now, Candida albicans is nothing but yeast, a yeast infection, and you can see here on the right side of the panel that there is a correlation between the amount of amyloid beta protein present in the Alzheimer's brain and the inhibition of the Candida albicans replication rate. So this then would suggest that in some way, perhaps, amyloid beta protein has an effect on inhibiting organisms from replicating and doing their thing as they're used to do, just by their presence. Now, in the next slide, we dig into some more detail here. This is another example. This is a bacterium, E. faecalis, which was pre-incubated with AP beta 42. Then the bacteria were extensively washed and then treated with an antibody to the amyloid beta peptide chain. And you can see that the bacteria here are labeled with the antibody to the amyloid beta protein, indicating that A beta bind tightly to the surface of these bacteria. To strengthen the case, in the next slide, this was an experiment that was conducted in 2014, so only a couple of years back, where chlamydia pneumonia was intranasally given to animals, and their brains were examined sometime later, and you can see here that the infection through the blood-brain barrier, that indicating the intranasal approach, which is a way through the blood-brain barrier, results into a deposition of anti-amyloid beta peptide 1 to 42 in these animals that are transgenic for this protein. So you need two factors. You need the transgene being ready to express the 142 alpha beta chain, and you need an infection with a bacteria in the brain, and as a result you get staining with an antibody against the amyloid protein. So there seems to be a direct link between an infectious agent in the brain and the recruitment of the amyloid protein to those cells which harbor the infection. The next slide. Now we come to part 3 as another essential component to make the story coherent, and that is the blood-brain barrier. The brain is a very privileged organ. Things don't get in, many drugs don't get in. It's being carefully protected from the outside world by so-called parasites, special structures in the blood-brain barrier. And as it turns out, our blood-brain barrier tends to slowly break down, as many other organs are in the body, but the location is quite specific. namely around the human hippocampus. So this is very recently published work in 2015. This is work done at the California Institute of Technology and USC in Southern California. The key author here is Dr. Slocovich, of whom you will hear later in a brief video where he and his postdocs explain the rationale and the methodology for this work. In the next slide, you see some of their findings. So post-mortem tissue analyses indicate that the blood-brain barrier in Alzheimer's disease is breaking down. In this study, they show an age-dependent blood-brain barrier breakdown in the hippocampus. And they believe that this is an event early in the progression to full-blown Alzheimer's disease. Next slide. This is now a video which I allow you to watch. It's quite intriguing how these researchers describe this, and I expect you back at the other side of the video, in which we will analyze this in more detail.
The blood-brain barrier limits entry of potentially toxic blood-derived products into the brain, as well as cells and pathogens. Recent experimental studies have shown when the blood-brain barrier is damaged, these products enter the brain, which can lead to neuronal injury, dysfunction, and neuronal degeneration. Human postmortem studies have shown that blood-brain barrier is damaged in Alzheimer's disease. However, the timing of blood-brain barrier damage is not known. Using an advanced MRI protocol, we show that during normal aging, there is an age-dependent blood-brain barrier breakdown in the hippocampus, a region involved in memory and learning. This blood-brain barrier breakdown in the hippocampus is worsened in people with mild dementia or mild cognitive impairment. Interestingly, there were no changes in any other brain regions in the blood-brain barrier permeability. We also found by CSF analysis that there is an injury to pericytes, cells which keep integrity of the blood-brain barrier. This study indicated that blood-brain barrier damage in the living human brain in the hippocampus is one of the very early events and may contribute eventually to cognitive impairment and dementia.
We use a novel advanced dynamic contrast enhanced MRI, DC-MRI protocol and post-processing analysis with improved spatial resolution and signal-to-noise ratio. Our method allows for the first time simultaneous measurement of the blood-brain barrier K-trans permeability in different white and gray matter regions in the living human brain. For example, we were able to characterize the blood-brain barrier K-trans values not only in brain regions as large as the entire hippocampus, but also in the hippocampal subfields. The arterial input function, AIF, was determined in each individual, which is really important, particularly if the studied population diverges by age, as changes in blood volume and flow may affect the AIF and the final K-trans measurements. For calculations of K-trans values, we used a modified path-like analysis. And finally, the post-processing imaging analysis was performed using our new software, Rocketship.
In this study, we developed a very sensitive assay to measure regional blood-brain barrier permeability in the living human brain. We injected gadolinium, a chemical that helps visualizing the blood-brain barrier integrity in 64 subjects of different ages and with different level of cognition impairment, with no and with mild cognition impairment. We performed neuropsychological tests to determine the level of dementia.
These results suggested that the hippocampus, the brain memory center, was the only region where the barrier broke down, becoming 41% more permeable with age in subjects who showed no signs of cognitive impairment or dementia. These barriers were in average 24% leakier in older subjects who showed early signs of dementia on neuropsychological tests. In some subregions of the hippocampus, such as CA1, they were even more than 50% leakier. Our data suggests that early vascular leakage in the aging human brain begins in the hippocampus and worsens with mild cognitive impairment or dementia.
We also determined cerebrospinal fluid biomarkers of blood-brain barrier injury and of different cell types within the neurovascular unit, including endothelial cells, pericytes, inflammatory response, amyloid beta, and tau. Cerebrospinal fluid taken from subjects who showed signs of dementia had a 115% increase in soluble platelet-derived growth factor receptor beta, a protein known to control pericyte growth and survival that we show is released during various types of pericyte injury. No changes were found in endothelial and inflammatory markers, amyloid beta, or phosphorylated tau.
Experiments in genetically engineered mice confirm the idea that soluble PDGFR-beta is released into the CSF during damage to pericytes, indicating that damage to pericytes may be a critical step controlling the vascular integrity in models of small vessel disease and Alzheimer's disease, leading to neurodegeneration. Our data suggests that pericytes may be an important target for developing treatments to avoid dementia.
To prevent dementias, we may need to come up with the ways to reseal the blood-brain barrier. But before we do that, we also need to understand how genetic risk factors, vascular risk factors, and environment influence the blood-brain barrier in dementias and Alzheimer's disease. It would be also important to understand how blood-brain barrier breakdown influences cerebral blood flow and white matter connectivity and structural changes.
Okay, welcome back. So now you have watched the video, and as you know, this group at USC is homing in on the hippocampus here. The structure here, and you can see the different subregions, the CA1, CA3, and the dentate gyrus. And the researchers employed a methodology of advanced MRI, functional MRI scanning, which allowed them to test the leakiness of the blood-brain barrier. The next slide shows you some of the findings that they made and the technology that they employed. So they looked at the SPDGR beta protein, which is a big protein derived from the parasite. This should not be in the spinal fluid at all, but when the blood-brain barrier breaks down, it will be found in the spinal fluid. Next slide shows you a strain of mouse which has a deficiency in the parasite, in those cells which are instrumental in the blood-brain barrier. And you can see also that in a mouse model of Alzheimer's disease, you also get a leaky blood-brain barrier and the release of certain big molecules. The next slide. Again, you can see here where the parasite is located. There are different risk factors such as vascular risk factors, genetic risk factors, and environmental risk factors, which will lead to a compromise in the blood-brain barrier, which then allows the entry of toxins and perhaps infectious agents into the brain, which then are triggering the response of the amyloid protein. Next slide. So here now is the paper. This is part 4 of our talk. Amyloid beta peptide protects against microbial infection in mouse and worm models of Alzheimer's disease. Let's look at the next slide, which summarizes it. So amyloid beta expression protects against fungal and bacteria infections in the mouse. They show that the amyloid oligomerization, a behavior traditionally viewed as intrinsically pathological, may be necessary for the antimicrobial activities of the peptide. So the intriguing possibility then exists that beta amyloid may play a protective role in innate immunity, and infectious or sterile inflammatory stimuli may drive amyloidosis. Next slide shows you what beta peptide can do to a candida albicans here, and you can see there are these fibrils attached to the organism which become more and more extensive and basically precipitate the organism out of solution. In the next slide is an elaboration of this, how amyloid fibrils can interact with the organism. And the next slide. So this experiment employed a transgenic mouse, 5XFAD, which is normally negative for amyloid deposits, but it's kind of like a loaded gun. If the right stimulus is there, then the amyloid beta peptide can be produced in bulk rather quickly. So this is the setup. The mice were then infected with various organisms, and the response of this beta amyloid system was assayed. And next slide, you can see what happens. This is a salmonella infection into the brain. You can see the salmonella stain in red. You can see the alpha amyloid beta in green. And the most interesting part here is the overlay. And when you see a color change from red and green to yellow, you have total overlap of the two signals. So here you have the beta amyloid coding the infectious salmonella present in the brain. The time course is extremely fast. It only takes about 48 hours. So you have a rapid induction of beta amyloid release from these neurons from a transgenic mouse carrying the defective beta amyloid gene from humans. Normally this is dormant, but in an infectious context you have a very rapid activation of beta amyloid release and an interaction with the infective agent, ostensibly in an attempt to neutralize the infection in the brain. The next slide shows you a video that shows you in a rotating way the direct relationship of the alpha beta amyloid and the salmonella and how they are totally superimposed. And go into the next slide. One final corollary to all of this is the ApoE4 connection. Now ApoE4 is one of the alleles of the apolipoprotein E gene. We all have this gene. It's an essential protein in the trafficking of lipids, cholesterol, and others. There are a number of alleles, namely 4. The ApoE4 allele is quite common in the population, and if you are unlucky enough to have one allele, you have a 5-fold increased risk of Alzheimer's disease, and if you have both alleles and you're homozygous for the ApoE4 gene, you have a 20-fold increase for Alzheimer's disease. What is the mechanism and how does it relate to what I have told you before? Next slide. So here is a cartoon which shows you the gene, and you can see the different haplotypes and the structure of the gene as it occurs in man. And again, a meta-analysis demonstrates significant association between the E4 allele of ApoE4 and Alzheimer's disease. The next slide shows you what ApoE4 can do. It is in fact involved in the trafficking of lipoproteins, as well as perhaps of some alpha-beta oligomers. This is the main pathway here. You can see that ApoE4 can complex with microglia and be involved in the removal of the naturally occurring oligomers of the amyloid alpha beta. So it's part of the transport chain that is supposed to remove amyloid from the brain. However, there is another and perhaps darker side to ApoE4 in the next slide, and that has to do with its effect on the blood-brain barrier. It appears as though if you have the ApoE4 allele, something goes wrong with your parasite functioning. Your blood-brain barrier becomes compromised. And this has been tracked down to a pro-inflammatory type A nuclear factor KB matrix metalloproteinase 9 pathway in parasites. So I know your eyes are glazing over, but it's a well-defined chain that is important in protecting the health of parasites. In the next slide. Here is what you see when you have a leaky blood-brain barrier, and you see here that stuff is leaking out of the microvessels. CYP-A here is compromised. If you add cyclosporin, which is an inhibitor of this protein, you can restore the integrity of the blood-brain barrier. The next slide. So parasites are known to play a key role in the development of cerebral microcirculation and protecting this microcirculation. Next slide. And now I'd like for you to watch a brief video in which the researchers describe how they went about showing the impaired blood-brain barrier as an age-related phenomenon in normal aging and likely in Alzheimer's disease as well.
I'm Beca Zlokovic, neuroscientist in Rochester. I would like to introduce our paper on parasites that is coming out in this issue of Neuron. Neurovascular unit is comprised of vascular cells, endothelial, and pericytes, glia, such as astrocytes and microglia, and neurons. The exact role of parasites in the neurovascular unit in the adult brain and during brain aging remains elusive. However, it is known that parasites play a key role in the development of cerebral microcirculation. Using two different adult viable parasite-deficient mouse strains with variable degrees of parasite loss, we have investigated whether parasite loss in the adult brain and during aging can influence brain capillary density, resting cerebral blood flow responses to brain activation, and blood-brain barrier integrity to serum proteins and blood-derived, potentially cytotoxic and neurotoxic molecules. We have also studied the effects of an age-dependent parasite loss and the resulting hemodynamic disturbances on neuronal structure and function and the onset of neuroinflammation. Mouse lines that we used in these studies express PDGFR-beta exclusively on parasites and not on other cell types in the CNS.
Hello, my name is Robert Bell. I am a PhD graduate student in the Zlokovic Laboratory.
After initially confirming a significant reduction of parasites in 1-month-old PDGF receptor beta heterozygous mice, we found that the reductions of parasites progressed as the animals aged. We then asked the question, how does a loss in brain parasites affect the cerebrovascular system? Using in vivo multiphoton microscopy as shown here, we were able to generate 500 micron thick Z-stack fluorescent angiograms after a tail vein injection of 2 million Dalton fluorescein conjugated dextrin. We found a significant reduction in the length of perfused capillaries, which corresponded to reduced cerebral blood flow measured by autoradiography, beginning at 1 month of age in parasite-deficient mice. Using hypoxypropimidazole, we confirmed that the reductions in cerebral microcirculation lead to severe hypoxic changes in the brains of PDGF receptor beta heterozygous and F7 mice. Next, to determine if there is vascular permeability and blood-brain barrier breakdown present in parasite-deficient mice, we utilized time-lapse imaging of a low molecular weight 40,000 Dalton tetramethylrhodamine conjugated dextrin. As you can see, in normal wild-type littermate controls, the dextrin remains within the blood vessels over a 30-minute time period. However, there was significant leakage of the dextrin from the blood vessels in parasite-deficient mice. The vascular permeability in parasite-deficient mice was also confirmed using a non-invasive fluorometric technique.
My colleague will now introduce how these vascular changes may contribute to neurodegenerative processes.
Hello, my name is Ethan Winkler, and I'm an MD PhD student in Dr. Zlokovic's laboratory. As Bob just explained, we discovered two parallel pathways, reductions in cerebral microcirculation and blood-brain barrier breakdown, which may potentially lead to neurodegeneration independently or in synergy. To confirm this hypothesis, we first examined the presence of several serum-derived neurotoxic and vasculotoxic proteins, normally excluded from the brain by an intact blood-brain barrier. As seen here, there was an age-dependent increase in perivascular and parenchymal fibrin deposits that correlated well with reductions in parasite coverage. Furthermore, as this image demonstrates, fibrin was observed to leak from small cerebral capillary segments lacking parasite coverage, supporting the notion that parasites play an integral role in maintaining the adult blood-brain barrier. To evaluate whether toxic accumulation of serum-derived proteins and hypoxia could lead to a secondary vascular-mediated neurodegeneration, we analyzed neuronal structure and function with several techniques. As seen here, Golgi-Cox histologic analysis revealed a significant progressive loss of dendritic spines in the PDGFR-beta heterozygous mice at 8 and 16 months of age. Similarly, hematoxylin and eosin staining noted significant structural abnormalities in the CA1 hippocampal subfield at the same time points. In agreement with these results, novel object location and recognition behavioral testing showed progressive age-dependent impairments. Significantly, no neuronal structural or functional deficits measured by behavioral testing or extracellular electrophysiologic recordings were noted in 1-month heterozygous animals, an age in which a pronounced vascular phenotype is already present. Therefore, this suggests that neuronal degenerative changes develop following a primary vascular insult in this model. We now return to Dr. Slockovic for some concluding remarks.
Now I want to summarize what my colleagues were showing you. We showed that deficient PDGFR-beta signaling results in pericyte degeneration in the adult brain and aging, leading to brain vascular damage by 2 parallel pathways. One, a reduction in brain microcirculation, causing diminished brain capillary perfusion, cerebral blood flow, and blood flow responses to brain activation, which leads to chronic perfusion stress and hypoxia. And second pathway, blood-brain barrier breakdown associated with brain accumulation of serum proteins and several vascular toxic and neurotoxic macromolecules, leading to secondary neuronal degenerative changes. We show that age-dependent vascular damage in pericyte deficient mice precedes neuronal degenerative changes, learning and memory impairment, and the neuroinflammatory response. Thus, pericytes control key neurovascular functions that are necessary for proper neuronal structure and function, and pericytes loss result in a progressive age-dependent vascular-mediated neurodegeneration. Our new vascular concept of neurodegeneration will have broader implications for our understanding of neurological disorders associated with perfusion stress and BBB compromise, such as Alzheimer's and ALS. It might guide new therapeutic approaches for neurological disorder directed at brain pericytes.
So here is a bunch of references that document the blood-brain barrier. These, of course, are available to the aficionados and those of you who subscribe to the enhanced content of this course. So that was a revised version and slightly enhanced version of our talk on is Alzheimer's disease an infectious illness. We need to explain, why does Alzheimer's disease occur in the 70s, 80s, and 90s and not earlier? And it may be because the blood-brain barrier does not break down until later in life. On occasion, Alzheimer's disease occurs much earlier in life, but then it's uniformly caused by a genetic defect in either the amyloid precursor protein or those enzymes which liberate the A-beta protein in excessive quantities in the brain. In those cases, of course, the illness begins in the 50s and 60s. So the story I told you today about the inflammatory and infectious component of Alzheimer's disease may provide at least part of an explanation as to why the onset of Alzheimer's disease occurs so late. Thank you for your attention, and there will, of course, be additional talks on Alzheimer's disease, which is a major topic in psychiatry and neurology. See you soon.